actually driving the third wave, which is thousands all the way up to millions. And then the mega, which is all the way up to a million cells and up to 96 samples in a single run. Yeah. Oh, you can't hear me on Zoom? Okay. It says it's on. We had all the time. Does this work better? Does that work? Okay, I'll keep it on me. Appreciate it. Thank you guys. So some of the real benefits of Parse, especially for people who are either coming to us for their first ever like single cell sequencing project, or those who are coming from other technologies, is really the fixation. So in the top right, our proprietary fixation allows you to fix and store samples for up to 6 months and then run them all at the same time to remove batch effect. We're also quite scalable. So really, as your project moves forward in scope, we could move with you. There's no instrument purchase, which is probably one of the biggest benefits, is that there's no service contract. We don't take up bench space, and everything you need to run the assay either comes within the kit or you already have on your benchtop. And then lastly, we're quite sensitive, and I'll show a couple slides on that in a second here. So then this is the overall workflow, right? You would start with your cells or nuclei. We have a fixation kit for either. You would then fix them. You would then do the library prep, and then you would sequence on an Illumina readout and then do the data analysis and visualization. And so just to show you how we are kind of different than conventional single cell seq, I'll also see if I can move that. In typical like drop seq kind of methods, you would be trying to get a cell and a barcode within the same oil immersion, and then you would lyse those cells and barcode the transcripts. And the problem with that is when you lyse those cells, you're stressing out the cell and you're losing potentially important biology. And so what Parse does that's different is we actually use the cell itself as a compartment. And what that means is you're not losing potential biology by lysing the cell, and you're also allowing you to use larger cell types than you would normally be allowed to use, because we're not trying to fit it into a microfluidic device. You're using cardiomyocytes. If you're using plasmablasts, other large cell types, or even bacteria is something that we can sometimes work with as well, then that's something that you can now do with our technology. So then the overall workflow, right? So the fixation takes about 30 minutes. You would fix your samples. You would then parse or split them out into the 96 well plate. Each well has a well-specific barcode represented by dark blue in this diagram. From there, you would repool your samples and split them out into another 96 well plate. This time they would get another random barcode. You then repeat this process a third time, and in the fourth step, instead of going into a fourth plate, you would go into 16 sub-libraries where they get affixed with a standard Illumina indexer. And I should mention that these are random hexamers, so you are getting 5' or 3' information instead of just 3' information. So then you can imagine, because we're doing 96 by 96 by 96 by 16, that you end up getting 14 million unique barcoding combinations, thereby making it relatively unlikely that any two cells are going to have the same barcoding combination. Another benefit of this technology is actually back on the previous slide, that fourth and final step. If you take a small aliquot of those fixed cells and run them, on an IC, you can determine the optimal reads for your sample and genes, and then you can run with confidence on a NovaSeq using that information for your larger cell sample set. So then going back to the doublet rates, as I mentioned before, it's relatively unlikely that any two cells will have the same barcode. And so what that means is we have an extremely low doublet rate. And so Parse did an experiment to determine what our doublet rates were by mixing mouse cells and human cells. And whenever you would have both in one, you would know that was a mixed doublet. And so what we found was that our doublet rates were about 3 times lower than other droplet-based methods, right? So if you're seeing 10 to 15% on whatever method you're currently using, with Parse, you'd be seeing 3 to 5%. And then we don't have any bias in cell type recovery. So this diagram here just shows you very clearly, when you comparison purple to flow cytometry, the results across different cells is very consistent, and even picking up rare cell types like plasmablasts. And then this just shows some data from our million cell experiment, just showing you that, if you wanted to run 1 million cells with us, it's very doable. So this was an experiment we did with 12 healthy donors, and 12 with type 1 diabetes. And you can see they used 40,000 cells per sample to create the following UMAP. Another benefit that I hear from researchers every day is that with Parse, it's a much more flexible workflow than you might be used to. So if you're used to having to stay in lab till 3 in the morning, or if you come in the morning and you're prepping your samples and you notice that they're starting to die as you go towards the afternoon, you're kind of running against the clock. Because with Parse, you've already fixed your samples, that eliminates that kind of that stress and speed requirement. We also have many steps in our protocol where you can pause and actually walk away from the bench or do a QC step. And so what the Allen Institute for Neuroscience in Seattle did was they took 3 different cell lines, 2 different protocols, and 5 different experiments, treated those all as 48 separate samples, and actually did it in one single run. And then this is the data that they got from that. As you can imagine, with current technologies outside of Parse, it's very difficult to do more than 8 or maybe 16 samples in a single run. And so really the benefit of Parse is that scalability. Another benefit is our unmatched gene detection. So you can see in this diagram here, on the left represents Parse in purple, gray on the right represents a droplet-based competitor. You can see that we only needed about, oh, sorry, I should draw the line. You can see from this line here, we only needed about 20,000 reads to get the same data that the competitor needed 40,000 reads. And so what that means for you is you can either use fewer reads and save the cost, or do a deeper dive and get more data for the same amount of cost. We also have we're sample agnostic. So we have a wide variety of flexibility across sample types. If you do not see your sample type on this list, there is a really good chance that someone else has actually used it. We just don't have a publication on it yet, or they haven't published data. So like I said, if you have a sample type that's rare or challenging, there's a chance it'll work on Parse. And then this is our long-term stability study, just showing you that if you were to fix your samples today and run them today, or if we were to fix them, throw them in the freezer, run them 6 months from now, you would still get 97% of the same data 6 months down the line. One final benefit that comes from the fixation is that if you had a large number of samples, like maybe like 1 million cells, you could run a small aliquot on our mini kit to determine sequencing saturation for your sample type. That way, you're not going beyond sequencing saturation unnecessarily. This is another way that you can save costs on sequencing. So then getting into a little bit about the barcoding and fixation. So all of our solutions have pretty much the same workflow. It's a 30-minute fix. You could then hand off those samples to the core where they're very familiar with doing the barcoding and library prep. And then it's just run on an Illumina instrument, and then you can do the data analysis. And so, one of the benefits, especially if you are to win our core grant competition here, then you could do the fixation since you know your samples the best, and they can do the library prep since that's their forte. And then just a really quick couple slides about data analysis. So basically you start from FASTQ files and you produce an experimental summary. We also give you a cell by gene count matrix. This pipeline can be downloaded and run locally. There's also an Amazon Web Service option. And then you can export this into Seurat or ScanPy as well. And so this is a picture of what our experimental summary looks like. As you can see, it gives you all the relevant information like cell number, sequencing saturation, and then transcript and gene detection. And then you can also create heat maps and scatter plots like the ones shown here on the right. And then lastly, we really want you to have success in this. So whether you're a core grant winner of this program or if you're running Parse on your own, we always schedule an hour-long training with you just to walk you through the protocol and make sure you have success. And then in theory, my colleague, John, is going to talk about, I see him popping on here, is going to talk about the core grant program. John, do you want to test the vocal?
Yeah,
here?
Thanks, Nate. Can you hear me on that side?
Yeah, we can hear you great.
Perfect. So thank Nate and the University of Florida for organizing everything. And sorry for some of the hiccups we had around the technology, but that's just the nature of the world today. So I appreciate it. Today, I'm just going to spend a couple minutes talking about the grant program. I suspect many of you showed up to hear about what this can look like to facilitate your research. And I'm quite excited about this program, and I think you will be too. So this is a there you go. This is a program that was put together for very prestigious universities and core labs to really push projects forward. So we wanted to not just show you a presentation of what this could mean using other people's data, but educate through demonstration. So giving this product to a University of Florida researcher in a way that allows them to answer an important scientific question and also communicate that to the wider audience. And not only just at University of Florida, but widely, we hope, actually. So the project here enables a single cell project for a University of Florida researcher. So anyone at the University of Florida is eligible to apply. It is a very simple process. We try to make this quite easy to participate in. We are not going to be judging you on, say, how well you can write an abstract. We're looking for projects that meet a few criteria. And the main purpose of this entire project is to generate those presentation-worthy results. So this is, the prize package here is not, say, enough to run 1 or 2 samples and just sort of, get enough to test out the technology. This is actually designed to answer questions. So if you go one more slide here. As Nate goes there, I just want to reiterate kind of that when we're putting this program together, it is a fully vetted kit. We've been working with the University of Florida core here already to help. And so there's a number of pieces that we're bringing together that we're supplying. And then there's also pieces that your core has donated as well. And so this is a partnership. We see this as a partnership, and we'll be working over time to make sure that everyone's successful. So it's important to just lay out exactly what these prizes are. What you'd be getting is the Evercode WT kit. So as Nate presented, we have 3 different sizes of kits. There's the Evercode WT Mini, which is up to 10,000 cells. There's the Evercode WT, which is up to 100,000 cells. And there's the Evercode WT Mega, which is up to 1 million cells. This kit is the 100,000 cell kit, which is a lot of cells to get data on. We're also providing 2 fixation kits. So these could be for either cells or nuclei, depending on your experiment. It supports up to 8 samples. And so it's important that we keep this well scoped in terms of sample size. Much more than this, the way our technology works is that 8 samples is divided into the 100,000 cells. So as you go up in sample size, you reduce the number of cells per sample. We found that 8 samples for these types of experiments not only is a good way to work with current experimental designs, but also give you tons of data per sample in a way that can really support that presentation side. You can submit an abstract, you can submit a project that's either cell or nuclei based. We do not make a preference. Everyone will be getting data analysis software. So if you get access to our technology, you get the software that goes with it. Nate presented a few slides on that. It really is designed to help you answer a lot of the questions that you may be answering. We also provide that software can output, say, gene matrix files if you had some third-party tools you wanted to use as well. There's plenty of training that will go around. So we don't see this as an opportunity to just train 1 or 2 people. Whoever wants to be part of this training can be. So we'll be training the core lab on a few of the parts that they'll be executing. We'll be training the researcher on the parts that they'll be executing, but everyone's able to participate in all aspects of this, not only just in the assay prep, but the data analysis side as well. This program, we don't do tons of these. We are really picking just a few institutions, and that's because we invest a lot in them. And so we're looking to make sure that everyone is incredibly well supported through this entire process. This may be your first time running our technology. This may be your first time running single cell. We want to make sure that you get every question you might have answered and you feel well supported and sort of that warm blanket of Parse throughout the entire process. The core lab is donating their services. So they will be doing barcoding and library prep, sequencing, and initial data analysis. So that is a major, I mean, first of all, I think you guys like them as much as we do. You have a fantastic core there, and they are bringing forward that support to help you with your project. This 100,000 cell 8 sample experiment will also go on an S4 lane of sequencing. So all told, you get a wealthy amount of data that you can use to support a project, support a research question, write a paper, do a poster, etc. This you will not be short of data. You go one more, there you go, thank you. This project is split in 2 ways. So the researcher will get the fixation kit. So there's 2 parts to this. There's the fixation, and then there's the barcoding and library prep kits. So the fixation kits go directly to the researcher. The researcher will probably also be doing, in biological data analysis. So the core lab will do the data analysis to make sure that you can get your results, make sure that the run looks good, those types of things. They may work with you on some of those higher level biology questions as well, but primarily we expect the researcher will be doing some of that. They will also do the fixation. The core side, so after your samples are fixed, you just bring them over to the core. The core will be doing barcoding and library prep. So they'll be doing the entire rest of the process, the sequencing and that initial data analysis. And so they'll get the WT kit. Now, again, you can be trained on whatever you want to be trained on and see it in action if you'd like. One more slide, please. So how do you participate? I think this is the part that if you've paid attention to what, you've patiently waited for me to get to the meat of the talk, that's where we're at right now. It's how do you apply? Well, there's a landing page that you'll be getting a link to. Some of you have already been on here. Others may come on shortly. You'll get a link to this, but there will be an area in the purple box there. Up till now, it was where you may be able to register for this program. You'll also now, that'll turn into an abstract submission form. So we're looking you to just tell us a little bit about how you want to use this kit. I'm not looking for you to write, a 5-page grant. This is like less than 300 words. We're not judging you on if you got, how close you got to 300 words. We just want to know what you're studying and how you want to use these samples, and also how it fits in terms of evaluation against the rest of the abstract submission. So how we'll be looking at these. And lastly, it's important to note, when you submit this, you sort of agree that once all the results are available, we're going to be able to use these for marketing efforts. And the reason I mention this is, if you're writing something that, maybe it's on a defense project or something where you're never going to be able to talk about these results, it's probably not going to be a good fit for this program. We do ask that when we generate results, we are sharing them with the world, and that's part of the emphasis of this program. There's 5 areas where we really look against these abstract submissions, and a well-written abstract will cover all 5 of these. Doesn't mean you have to, but that's kind of how we're going to be evaluating. So we'll be looking at, how ready is this project? Can you be, are you going to wait 2 years for your samples to be ready, or are they going to be ready in 2 weeks? That type of thing. How well fit your experiment is to the Evercode technology. If it's, some projects just naturally fit better with this. Are you willing to present these results when they're ready? And are you willing to be recognized as our winner? So we are going to do everything we can to really glorify your work. And we just want to make sure that you're willing to be glorified. And then lastly, is the project feasible? We do often like to fuel projects that we're just not sure if they're feasible, but oftentimes for these programs, we also want to see early successes, and so more feasible projects tend to be weighted a little bit higher than big moonshot projects. Another slide, please. So on a deadline, on a timeline perspective, we are talking today, July 14th. By tomorrow, that website will be updated with a recording of today's talk, as well as the application form. Next Friday, so we're only talking about a week now, at 11:59 p.m. on Friday, because we know everyone loves Friday night deadlines, will be the deadline for abstract submissions. Now, that's when abstracts will close next Friday evening. We'll be spending the next week reading through everything, thinking about them, talking about them. And when I say we, I mean the Parse team. So your internal core is not selecting the winner. Don't go yell at them if you didn't win or whatever. That's completely on us. We'll be notifying and working with all the abstract submitters over the following week. And then the week of, or the August 4th right now, if not earlier, we plan on announcing that winner on social media. So it's a very rapid timeline. You're not going to be submitting an abstract and waiting 6 months for approval. Everything happens very, very quick. Once this is gone, once we've sort of kicked off this project, we are looking to do quarterly check-ins, both with the winner and the core. We do like to just keep in touch and make sure that your project is running well, any problems you may have. We also see this as these winners are fantastic ways for us to learn more about our product as well. And so these quarterly check-ins are really a way to inform us how things are going, but also let you know what opportunities there might be for you to share your science with the world. And so it's a conversation constantly, and we just like to revisit it. With that, I think I just want to say thank you, and happy to answer any questions, both on the program or Nate on the technology or the core on anything else. So thank you.
So it is a single cell. It's just a pooled kind of subset of the samples that you put into the actual assay. So in 3 formats, you have a couple of options. In the smaller format, you could do up to 12 samples. At the very end, the 2 sub-libraries are going to be a mix of all of those samples together, but they are indexed with a UMI for the sample from the first plate, and then they get indexed for the RNA transcripts to the second and third plate. So the barcoding actually will delineate back to the actual sample, the single cell from that sample, and delineate it by row, which is then at the end create a mixed homogenate of all of those samples into the same cell. So you can end up with, depending on the format of how cells you want to target. So there's 10,000 kind of assay format, 100,000 cell assay format, and you can divide those up in any way you want. So take a smaller format and do 1 sample and get 10,000 cells plus all 12 lines. So you can do 2 or 6. It's very flexible into how many cells you want at the end and how much data you want. So that's the nice part about it. And how you been person you work, obviously, we had work here. We've done a lot of TEDx projects with the gene expression core and worked with a lot of people here doing their sample prep of those projects. The limiting factor is always running against the clock, right? You're running against viable cells, you're running against how fast you load them, and you're running against how many you can actually put into the assay. The thing I like a lot about Parse was it gets around a lot of those problems because you actually doesn't matter. You fix those cells and permeabilize them, and then the gene expression profile is frozen in time. You throw them in the freezer. So they don't get altered. There's no batch effect. And you can do long studies of microdosing or clinical trial samples across time and come back 6 months later, compile all those samples and run them in one assay altogether. So the data is more representative of where those cells were at that moment in time when you harvested them and were very healthy. So I think there's a huge benefit to the flexibility of the platform that's different than some of the droplet-based assays.
Great question. Thank you.
Well, again, it depends on how many cells you want to target. So the kits are designed with barcodes that will kind of target the maximum amount of cells that each kit's based on. So if you think about Google, right, it's targeting 10,000 cells. Now, yes, you could certainly overload that right, if you wanted to. Sorry. Okay. You could certainly overload that plate and double the loading if you wanted to maximize your cell retention and the barcoding on the way out. But at the end of the day, it's built to really efficiently barcode 10,000 cells. Now, you might get a little more, you might get a little less out of that. It depends upon how you load the samples into the plate, and the distribution is based on cell counting. So when you go into fixation, you're going to do a cell count, and what we do is kind of a mathematic multiplier, right? Much like sorting. We like you to go in with like 50,000 cells, for example, into fixation, because we know there'll be some retention through the plastic. You might have some cell loss coming out of the freezer. So we want to account for that, so that when you go into loading the plates, you have a correct cell number of like, say, 25,000 cells per sample at a minimum. And that's what we would consider loading across however many wells you designate. So then it becomes a distribution of like, random 10,000 cells, 4,000 cells, 3,000 cells, however you randomly distribute them across the plate. But the barcoding efficiency is very good at capturing all of those cells. So each cell gets individually barcoded. And at that point, when you look at the data on the back end, even though they went through different wells in the beginning, you can trace them back to the same sample subset, if that makes sense. Yeah. I think for the Parse assay, it's somewhere around 85%. Am I wrong on that, John? The efficiency of barcoding,
Sorry, what was the question?
The efficiency of the barcoding, if you put in like 10,000 cells into a well, the actual efficiency of the true barcoding per well, per plate.
I honestly don't have that number off the top of my head.
Okay. I don't either. I think it's 85%, but I can definitely find out for you. It's fairly high. So, yes. Typically, if you put in good quality fixed cells, then you're going to see those come back out in the transcripts, and you'll see it delineate, right? You can see what transcript fraction, because in the pipeline, it basically kicks out anything that doesn't have the complete barcode set. So if a cell, exactly, it's already filtered out when you do the FASTQ file processing through the pipeline analysis, it filters out anything that doesn't have the complete set of all 4 barcodes. So if you have an incomplete barcode, say a cell that's partially broken, or barcodes that a cell that doesn't get fully barcoded, it will kick it out and not show it as a complete read. So it won't associate it with a full mRNA transcript. So there's a way you can actually see that, and you can look at that efficiency proportion. But we find that it's fairly low. If you go in, the key to it, again, with most single cell technologies is good quality cells going into fix, but it's easier because we have the time on our hands, right? You just take the cells out of culture, you take them straight out of the tissue digestion, you do a viability check, you count them, you fix them, and they're frozen in time. So it's not racing so much against the clock like you used to with 10X or Rhapsody or other single cell platforms. So there's more flexibility there. But yeah, it'll kick out anything that doesn't get efficiently barcoded. But I will find out the full percentage for you guys and let you know as a follow-up. So that's part of the ligation reaction. So there is an actual, each well only has 1 barcode. So as you go through the process, there isn't a way you can multiply barcode the same cells in the same well, because their barcodes are actually different in every single well in every one of those plates. And the ligation reaction actually creates a linker sequence between the first barcode and the second barcode. And as you move them through plate 1 to plate 2, the barcodes are different in each format. And then you carry that information when you're done with the sequencing run. You'll kick that information back out and you'll throw it into the FASTQ analysis, and then it reassociates all the information for you through the pipeline. So it's a very nice process where you don't worry about that. I give you a 10X. Yeah. Sometimes, depends on the species. So in general, it depends upon, like, for example, we see more of it in insects because insects have a high content of ribosomal RNA. So for example, we've run some Drosophila and some other insect studies. We see a higher percentage of some of the random or hexamers priming that ribosomal, but we also remove that information. We'll map it as multimer mapping in the pipeline, so you can see what percentage is ribosomal and what percentage is intact RNAs. For most of other organisms, we're not seeing that. So I think it's kind of just a species specific thing, as far as what we've seen already in the field and what they've generated in-house. So
It's generally not a problem.
no, I don't think that it is, to be honest with you, because we don't see it in a lot of mammalian stuff. We don't see it in a lot of human stuff. We're not seeing it in a lot of plant stuff currently. No, I don't actually have a good answer for that. Yeah. But I think it has to do something with the way that the chemistry reaction actually works. So,
Great question.
but that's a good question. Yeah. I can find out more information and give that to you guys. So do we have any questions? So, right. So again, same problem we always have with most single cell experiments, right? You're going to see some of that because inevitably you have cells that are cracked or cells that have died in your process, right? Two things that I do and I did before and I would recommend you do now is first thing is add some RNase inhibitor because it minimizes some of that. And the second is that a lot of that you're going to end up removing in your secondary data analysis as long as you're putting in not a ton of broken and damaged cells into the actual barcoding assay, you minimize that. So we don't see a lot of it if you're putting in intact permeabilized cells. And we have multiple steps in the actual assay where you visually have to do a cell count and a morphology check on the cells. So you're checking the quality of them before they go into fix, after they come out of fix, after they come out of the freezer, and when you're done with barcoding. So we know exactly how many cells went through the entire process, how many you started with, how many you ended with, and what their morphological characteristics look like as far as membrane integrity and if they're leaking cytosol or transcripts into the solution, because we can get a gauge on how much you're going to see in the sequence analysis later. But inevitably, you will still see some. But our goal is if we get live cells with a good viability, 85 to 90%, 75% or better, you do a little bit of a cleanup step, add some RNase inhibitors. The quality of what you get back is very minimal with broken transcripts. So I'm not seeing a lot of it in the data analysis sets that I'm looking at. If you handle the cells correctly on the front end, and the beauty of it is that it's easier to get the cells quickly out of digestion and into fixative than it is move them into, 10x and loading and like some of those complicated things that you got to do a lot of manipulation to get everything, the timing to work right. You don't have to worry about that here. So I think from my perspective, that is an advantage of Parse that I think makes it very different than other platforms and easier to use because it captures your gene expression really nicely and really clean. So. And we got a couple from the, yeah, are these?
They're just asking if we could repeat the question.
Oh, okay. Oh, okay. Okay. All right. Do we have any other questions from the people on Zoom? No, maybe not. Okay. And the other beauty of what Parse does, which is one of the reasons, from my perspective as a person who used to be here at UF, part of the biotech core, I was interested in coming into this company because I think they fulfill a need in the single cell space that we have a 10x. I have a Rhapsody in my lab, right? They fill a niche that the droplet-based competitors aren't filling. However, at the end of the day, I'm a very pragmatic person, right? Single cell is single cell. If you don't put good quality cells into it, you're not going to get good quality data out. But the beauty of it is it's much easier to get somebody and help you fix your optimization on the front end going into a fixed reaction than it is trying to get the timing right and all of the machinations right to get those cells in and out of 10x correctly, alive and intact. So it simplifies things on the front end. The other thing is from a support side, what my job and Colleen's job are, is that we're here to like help you through that whole thing, right? From the day you want to start the process, we'll talk about what kind of cells you have, what kind of protocols you're using for getting the cells out of culture or digesting them out of organs or tumor infiltrates or whatever your project looks like, and help you optimize every step of fixation so that we have really great quality cells going into barcoding. Because if we can do that, then everything else downstream is very, very easy mechanistically. It's just the front piece. And actually, that's not as hard as you would think, right? Having been a person who does a lot of sample prep, kind of my wheelhouse. So I've worked with a lot of weird cell types and optimized a lot of weird things. So it's something that I think Parse does really well, is that we're getting much more efficient in how we help people think about getting those samples into the fixed reaction and out of fixed reaction with great quality. Yeah. Well,
but yeah, I'm always happy to do project consultations or the quick meeting over Zoom or even in person today. But we're dramatically more affordable than a lot of the droplet-based competitors out there. But you know, the bacterial thing is one where we don't have an official protocol, right? So a lot, like we said, we're sample agnostic. We have had people use Parse successfully with bacteria using a SPLiT-seq publication that somebody came out with. So, you know, if you were to use that, we'd kind of give you that. We'd start you there with a guide. And I forgot the second part of this question.
oh, just asking if you could combine the bacteria and the cells together. Technically, yes. It would be kind of an off-label modification, but we are always, like, I'm a huge proponent of, like, I will jump off the bridge without a parachute with somebody. I actually really enjoy those kind of projects because I think there's a lot of success that we can get out of those, and I'm working on a couple of those actually right now. I don't have the data back from those yet, but they're in progress. So if you give me a couple of weeks, I can give you a better definitive answer on that. But for the bacterial piece itself, it's a modification in the fixation process. So it's not using the kit-based reagent, it's using a different formulation, slightly different, but it does work. So
Yeah.
yes, you can do it. It's just a modification of not our actual kit-based, more of the paper original paper. But
Yeah.
It's been done very successfully.
My point was, you know, there's a lot of technologies right now for single cell where if you have bacteria, it's not even an option at all. They'll just tell you we cannot do this. And with Parse, it is a potential option.
Discuss the challenge of extraction. Okay, so challenging of extracting cells from tissues depends on the cell type. So for Beth, I'm going to be honest, that depends on what you're working with. Kind of my area of expertise, if you don't know who I was, many years in the flow core here, I've probably done just about everything known to man, human, animal, vegetable, and other. So I have a lot of protocols. Parse has a ton of depth of field in this, where we've got great protocols if you don't have them. If you have a tissue type you're working with and you're struggling with, you can definitely reach out to me. Skeletal muscle. Oh yeah, got a great protocol for skeletal muscle. You can either work with the cells or with nuclei. We find that sometimes nuclei extraction is a little bit easier and a little bit better because the muscle cells are a little bigger. However, I have a great protocol for that. And if you want, if you email myself and Nate, I'm happy to share the protocol I have with you. No problem.
Great question. Thank you, Beth.
Thanks.
Thanks, everyone. Thank you, guys.
Thank you, Steve, for hosting. We appreciate it. Yeah.